membrane. Okay, so we talked about this a little bit before. The cell membrane, we also say, the plasma membrane. But guys, basically, this is based off of the fluid mosaic model, which basically says it's not rigid, it's not hard. Basically, it's very flexible. It's going to be able to move in and out. We're going to be able to have different substances go in and out of this through the different types of transport. That's what we're going to be talking about next week. And that's what this little diagram is showing you, is different parts of the different transport. Guys, in just a second, like these right here are the phospholipids. What's on the outside of the phospholipids right here? The hydrophobic heads. Sorry, hydrophilic heads. And these right here are going to be the hydrophobic tails. These are going to be nonpolar. The heads are going to be polar. Because what is our body mostly made up of? Water. Cells are mostly made up of water. So you want the hydrophilic, which is the water loving on the outsides. Okay, so I want y'all to look. It's really, really flexible. What part of the phospholipid allows it to be flexible?
The double.
Right. It's the double bond in there causing it to be what?
Bent.
Bent shape. So is that saturated or unsaturated?
Unsaturated.
It is called unsaturated. Okay, so when I talk about the cell membrane, guys, this is what basically I just said. The cell membrane, it's got the two layers of phospholipids. We've got the polar heads and the non-polar tails. The tails are on the inside. And guys, it causes this region of like water resistance. That's what allows certain things to actually go through the cell membrane and certain things not to go through it. So if it is something water-based, guys, once it hits that little non-polar tails, are they really going to be able to go through the rest of the cell membrane? Something water-based, they hit the non-polar tails, are they going to be able to continue going through it? No, they are not. And so this helps to waterproof the cell. It helps to keep us from losing too much of those substances. So in a little bit, we're going to talk about different types of channel proteins that allow the diffusion and osmosis to actually occur. All right, so guys, when you see this word amphipathic, it just means it has both properties, hydrophilic and hydrophobic. Now, we have these unsaturated bonds right here, that little double bond that causes that little leg to pop out. Guys, this is what allows it to not be so just in line, rigid. We want to have that fluidity. This right here, they're too closely packed together. Now, because of this, sometimes we'll also have a little cholesterol embedded in that also helps keeping it from getting too ordered, too structured, which we'll talk about here in just 1 second. All right, so are we good with this slide? We've already talked about this stuff. All right, so we're going to talk about the cell membrane, guys. We say that those phospholipids are semi-permeable. There's only some things that can actually go through the cell membrane. They can actually weasel their way through. It's going to be small little gases. Small little particles can actually weasel their way through and go to the other side. Now, if I am a hydrophobic, what's the word hydrophobic mean again? It is not like water, so it's going to be non-polar substance. It can actually weasel its way through these non-polar tails. So if it is a hydrophobic substance and it's small enough in size, it can actually go through. Certain small polar molecules can also, some of them can. But larger molecules, they have to go through your different channels, different proteins. All right, so if this right here are my cell membrane. Guys, you've got your different phospholipids. You have carbohydrate chains that come out. We'll talk about that here in just 1 second. You have proteins embedded in it. Some of these proteins have little channels for allowing some of these molecules to be able to go through from a high to low, high to low, high to low, high to low, high low. Remember that from freshman year? Because in diffusion and osmosis, we go with the concentration from high concentration to low concentration. High, low, high, low, high, low, high, high to low, high to low, high to low, high, low, because you're going down the concentration gradient. All right. So when we talk about the different proteins, guys, I just said a second ago, proteins can be embedded in that cell membrane. All right, so we've got two different types of proteins. These are the main names for them, peripheral and integral. Peripheral are going to just be temporarily attached, just like right here. They're just going to land on the outside of the surface. It's just going to temporarily to that surface. It's going to interact with them. It's going to be a lot of the signaling, some of the different communication is going to go on with like your insulin and your hormones. Okay, integral proteins, they integrate themselves into the cell membrane. And so these are going to be permanently attached. So they're integrating themselves into the cell membrane. These are peripheral, where they're just attached to the outside or like the inside of the cell, but basically the outside of the membrane. But integral is where they're integrating themselves in. These are permanent, and guys, these are going to be more like your channels, mostly for your channel proteins for transport. They could be used for some types of cell communication, which is actually one of the upcoming chapters. Okay, so this is more of what they look like. They're not going to be blocks, but this is more of what they're going to actually look like. It's going to be a protein chain. We've already talked several times about proteins. Remember proteins, we've got the primary, secondary, tertiary, and quaternary folding going on. So like right here, we've got, what would you call these? Are they integral or peripheral? These would be integral proteins because they are embedded in that cell membrane. Okay, so proteins can do all kinds of different things. They can have intercellular joinings. So like if I have one cell that gets really close to another cell, sometimes they will actually almost like collide and touch, and they can exchange things back and forth. We'll talk about that here in just 1 second. But they can exchange things back and forth from one protein to another, just direct from one cell to another cell. We can also have them where they have some enzymatic activity going on, like the lock and the key, where the substrate snaps into place, and either the substrate, two substrates combine to form one molecule, or like one molecule as the substrate locks into place and it's able to divide them into two. We do have them as your transport active passive. This is actually what we're going over on Tuesday. Cell-to-cell recognition. This is going to be some of your different cell communication. And so you have a carbohydrate chain that's attached to this protein for that cell communication. Okay, helping it to anchor in. We're going to talk about the cytoskeleton here in just one moment, and some of the exoskeleton. So it helps it to anchor into place into, say, in a one little area and not be fluid and moving all around. We can also use some of these proteins for signals. Guys, not only for, this is more of the cell communication. I'm sorry, let me backtrack. This is more of the cell communication. This is going to be like recognizing one cell to recognize what other type of cell it is, whether it's another heart cell, whether it's going to be another type of cell. It can also help with recognizing your cells versus somebody else's cells. So this comes into play whenever we're talking about like transplants. That's why you have to be a good match. Otherwise, if the cell cannot recognize another cell, they're going to think it's a foreign body and they're going to try to destroy it. Okay. So I talked a while ago about that cholesterol. Guys, the cholesterol will actually embed itself in between these different phospholipids. What is it trying to do if it embeds itself in right here? It's trying to help keep it more fluid. Guys, if I just had phospholipid, 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 it still gets too rigid in there. We want to have some of this cholesterol embedded in place. That way it helps keep it from being too rigid. It helps with that flexibility. And so, hey, what type of macromolecule is cholesterol? It is a type of lipid. All right, so plants do not have cholesterol in their cell membranes. So basically, they just help have more of the little legs kinking off to the side. They help have more of the saturated versus unsaturated fatty acids to help with that fluidity. For us humans and other animal kingdom organisms, we have that cholesterol embedded into the cell membrane to help increase fluidity. Now, organisms can alter the amount of fluidity that their cell membranes have. Like right here, if it's too hot, then cholesterol can help actually stick to phospholipids and pack them in closer and closer together. It makes the membrane a little bit more rigid and less permeable to the small membranes. If it's lower temperatures, then this cholesterol keeps those membranes, the cells from actually, the cell membranes from packing in as tight. So think about that whale. We were talking about the whale the other day versus the elephant. If I have the whale in colder temperatures, guys, think about things when they get cold. Don't things typically like shrink in and get smaller in size? Well, this helps to keep that from happening. It helps to keep that cell the size that it is and to help keep it nice and kind of fluid. And so we do have this to help maintain homeostasis. Okay, carbohydrates. I showed you a while ago, you've got those carbohydrate chains. They're going to be used for a couple of different things, mostly for cell recognition and for this communication. So sometimes they're embedded on top of a protein, sometimes they're just embedded in the cellular membrane. All right, so you have glycoproteins and glycolipids. Because what does the word glyco make you think of? Glucose, glycogen, glucose. All right, so these are going to be carbs are made up of glucoses. So glyco attached to a protein, or glyco attached just inside the actual phospholipids. All right, so your glycoproteins are attached onto a protein. Your glycolipids are just embedded in the cell membrane right by those phospholipids. All right, so that's what I was talking about a second ago. When we say cell recognition, guys, if you're getting a transplant, they have to see if you're going to be a good enough match. And what that means is that these glycolipids and glycoproteins, that they have to be able to recognize one another. Otherwise, if you're not a good enough match, your body's going to reject that transplant. And so, yeah, even then, you still need to make sure you take some type of immunosuppressant medication. That way, your body's not attacking itself, trying to kill the different cells in that new organ that you were just given. So, really, really important if you're doing transplants. So you definitely have to make sure that you have a good match. All right, so this is just kind of to recap. You've got your cell membrane right here, or we can also call it plasma membrane. What are these? Phospholipids. What is this? Cholesterol. What does it do? Makes it a little bit more flexible, and that's what it's showing right here. Hot versus cold environments. All right. Yeah, if it's hot, guys, think about things as they get hot, like plastic. Whenever it gets hot, what happens to that plastic?
It becomes more malleable.
It becomes a little bit more pliable, malleable, a little bit more pliable. And so we actually, because that cholesterol, if we have a little bit less, it helps to make it a little bit more rigid. That way, it will help to hold its shape. So we take some of the cholesterol out, so that way the phospholipids can actually line up a little easier. All right, and then you've got your glycolipids and your glycoproteins. So your glycolipids help with the cell recognition and to attach, to adhere to other surfaces, to other cells. Your glycoproteins are going to be linking of cells, giving the tissue some of its different strength. So, all right, let's talk about the cytoskeleton really fast. So cytoskeleton, this is the inside portion of the cell. So these are different things that help to give the cell its structure, its different shape, its different structure. It's kind of like, think of it as the bones of the cell. So like right here, it gives it, it's part of its structural framework. So we have different things that help with this. We always talk about microfilaments, microtubules. Guys, this is just things that are floating around, like inside of these walls. What do we have inside of these walls to help give it structure and support? You have like rebar. What else would you have inside these walls? Reinforcement 2x4s, like your studs. You've got all kinds of things. It's not just the wall itself. You have things embedded in to give it structure and support. And with the cell, it's the same way. You've got these different things floating around that help to give it more of its shape and to give it the support that it needs. All right, and so you kind of need to know, these right here. Now, eventually, some of these will start helping whenever we're splitting the cell apart, whenever we start talking about the spindle fibers and everything else. All right. So right here, your cilia and flagella are parts of your cytoskeleton that are going to extend out past the cell. We use them for mobility. So cilia are going to be those little hair-like extensions that we use for movement. Your flagella is going to be your tail that we use for movement. Cilia can help us also with some of the attachment if we need to, but it's mostly just the tiny little hairs for movement. All right, we also have something called the motor protein. So Your motor proteins are going to be found in different areas where it's trying to move part of a cell from one area to another. So these little feet right here is having some of the cell recognition. It's allowing it to attach on to certain parts within your body, helping to move your cells. All right. And like I said, we also use them for the different microtubules where the spindle fibers form to help pull the cell apart. That's where the centrioles and centrosomes come from. Okay, let's talk about the external cell structure. So we have things outside of the cell that also help to support us. We always talk about collagen. Collagen is going to be one of those different proteins that's going to be found outside of the cell, but it helps our cells. So plants are made of cellulose. Sorry, let's talk about cell walls first. Plants are made out of cellulose. Fungi. Where's some fungi? What are examples of the kingdom fungi? Mushrooms. That's the most prominent one. Sometimes people will say slime molds. It's mostly funguses, like your mushrooms. They have cell walls made out of chitin. So remember, these are both different types of carbohydrates. So they're just long polysaccharide chains. Okay, so that's what your cell wall is made up of. It's very, very rigid. It does not flex really at all. Now, on the outside, you have your cell wall. Just on the inside, you have your cell membrane. The cell membranes can actually detach from the cell wall and shrink. We did this when we were doing the onion cell osmosis. Y'all remember us doing that, where we put the different solutions on? You can actually see the cell walls the cell membranes detaching from the cell wall, and you can still see the shape of the cell wall, but you can see that whole cell like collapsing on the inside. We're actually going to do that lab next week too. Anyway, we've got this extracellular matrix. So guys, look at the curvature. You know that this is on the inside of the cell, so that's part of your cytoskeleton, like your microfilaments, microtubules. You've got your, what are these? Phospholipids. What about this?
Protein.
What type of protein? The integral proteins. And then you've got your extracellular matrix. These are just different types of proteins, such as that collagen, that help to give the support, a little bit of the cushioning, a little bit of scaffolding to help hold the cell together. The cells can actually adhere to this. This is what I was talking about a second ago. That some of those proteins can actually, on the cellular membrane, adhere to other substances, but they're going to adhere to part of this extracellular matrix. Guys, if we don't have enough collagen, if our collagen starts to go away, that's when we start to see a lot more of our rapid decay on our bodies, like through our skin. That's when we start to see a lot more of the aging going on. We need to have this extracellular matrix right here for our cells to be able to hold on to adhere. Once this starts to go away, that's when we start to see more of that aging starting to appear. Anyway, their cells just can't support themselves as well. So, okay, and this right here, this is called a cell junction. A while ago I showed how two cells can actually butt up right next to one another, and they can connect to one another and have some of the communication happening. And that's what's occurring here. So we can actually butt them up towards one another, and they can attach, and they can actually start to communicate back and forth. They can share different substances. They can communicate back and forth. We call those gap junctions. Basically opens like a channel. It's like a little, so this is a plant cell. With these little gap junctions, we're going to be able to have transport going back and forth through one plant cell to another plant cell. All right. And that's where I'm stopping for the day. All right, so we're going to go into the back. We have a lab to do plus a little worksheet. So it's actually a really easy day for the rest of this. It's the bubble lab. It's fun.
Bubbles.